This presentation will include an investigation of potential vaccine production routes for the manufacture of a Zika virus vaccine. Zika virus is a deadly single-stranded RNA flavivirus whose RNA codes for a polyprotein. That polyprotein is then cleaved into structural and non-structural proteins. The structure of Zika virus is shown here in Figure 1. The disease is primarily transmitted via mosquitoes and mother-to-child vertical transmission and can cause severe neurological and developmental complications in infants. Thus, the World Health Organization, WHO, has identified the disease as a major health risk, requiring further research and development to treat and prevent. This presentation will primarily focus on production routes for different types of potential Zika virus vaccines. Before discussing the production routes, first we'll go through a brief summary of how each vaccine works. We will primarily be discussing 4 types of vaccines: DNA, mRNA, live attenuated, and inactivated vaccines. DNA vaccines provide immunity by relying on injected plasmid DNA. The plasmid DNA gives our body instructions to produce an antigen of interest that would help provide immunity against infectious disease. Unfortunately, there have yet to be any DNA vaccines approved for human use. This is partly because the injected plasmid DNA would be integrated into host DNA, which is a cause of concern. Next, moving on to a more familiar face, mRNA vaccines. mRNA vaccines help create immunity against disease with a similar mechanism as the DNA vaccines. However, they use mRNA instead of DNA. Because they use mRNA, they do not integrate into the host cell genome and are broken down after they have encoded the antigen of interest. The front-runner COVID-19 vaccines produced by Moderna and Pfizer were both mRNA vaccines. With the DNA and mRNA vaccines, I've referred to an antigen of interest. The selection of which antigen to encode is important to ensure efficacy of the developed vaccines. Yellow fever and Japanese encephalitis are both part of the flavivirus family, the same family that Zika virus is part of. The vaccines for both those diseases express structural elements of their respective viruses, and studies have shown that the bulk of antibody response to Zika virus is in response to the envelope or E protein, which is also a structural protein. So a good target for an antigen of interest for DNA and mRNA vaccines could be the E protein. Next, there are also live attenuated vaccines, which involve injecting weakened pathogens into the body to induce immunity. A related example is the yellow fever vaccine. Finally, inactivated vaccines induce immunity by injecting pathogens incapable of replicating into the body. And relevant examples include Japanese encephalitis, influenza, and polio vaccines. Next, the following slides will include a discussion about the differences in production for each type of vaccine. Overall, there are 3 main phases of production for DNA-based vaccines: the upstream phase, the production phase, and the downstream phase. The goal of the upstream phase is to propagate the plasmid that encodes the antigen of interest. After propagation, a working cell bank will be selected and will be the source for any following plasmid production. Then, in the production phase, the cells with the plasmids are grown and fermented. In the last part, the downstream phase, the plasmid DNA needs to be retrieved from cells and purified. On this slide, we will be going over the upstream process and the production process. First, the plasmids are propagated with E. coli strains. Then, the plasmids are transformed into production cell lines, from which the resulting colonies are then screened to select a master cell bank, from which a working cell bank is selected. Now, moving on to the production phase, the cells are fermented and grown with fed batch reactors because they usually yield higher cell density and plasmid yield compared to the other reactor types. Then, the cells are harvested with either continuous centrifugation or tangential flow filtration. Now, moving to the downstream phase. There are multiple ways to retrieve and purify the plasmid DNA. In this case, we'll be referencing a patent used to manufacture multiple DNA-based vaccines, including influenza, HIV, and growth hormone-releasing hormone vaccines. First, the harvested cells will be lysed with alkaline lysis. Then, the plasmid DNA will be separated with filtration and anion exchange. Finally, any remaining impurities will be removed with hydrophobic interaction chromatography, which selects based on surface hydrophobicity. Then there will be another round of TFF. Overall, the DNA vaccine production process is limited because of the potential contamination of endotoxins, as well as the plasmid recovery process. While the vaccines have been tested to have lower immunogenicity and high safety as well as relatively simple production, there can be difficulties with the lysis steps during scale-up. In spite of the benefits, DNA vaccines may not be the best candidate for a potential Zika virus vaccine because there is yet to be a DNA-based vaccine approved for human use, which may delay manufacture due to lack of existing infrastructure and large-scale process development. Live attenuated and inactivated viral vaccines follow a similar sequence of steps, with inactivated virus vaccines requiring an additional step at the end of the process to deactivate the virus. Since yellow fever vaccines are in the same family as Zika virus vaccines, production of yellow fever vaccines could model potential production of live attenuated or inactivated Zika virus vaccines. First, according to WHO recommendations, a certified substrain of the virus is grown in a specific pathogen-free flock. From there, a master seed lot is selected, and then a working seed lot is chosen from the master seed lot. Seed lots are well-characterized viral suspensions that are uniform to other batches, which are processed at the same time. From there, the process proceeds with the working seed lot as the source of subsequent production. Testing for different characteristics and contamination is required at every step and can prove to be very time-consuming. Overall, the testing and handling can prove extremely labor-intensive, so this method is not ideal for large outbreaks. Then, the virions are recovered through either low-speed centrifugation or filtration. The incubation for inactivated viruses is the same, however, at the end of the process, virions will be deactivated with chemicals like formaldehyde. Both live attenuated and inactivated vaccine production methods are limited by the supply of embryonic chicken eggs. However, live attenuated viruses provide lifelong immunity with minimal doses, which could alleviate burdens on production. However, the egg supply and subsequent quality control could prove too time-consuming. Inactivated viral vaccines are especially appealing due to high volume of research already conducted for flu vaccines. However, as we know from getting yearly flu shots, inactivated viral vaccines do not offer strong immunity and could require booster shots, which would put further strain on production. To address the limited availability of chicken embryonic eggs, production of inactivated viral vaccines is shifting towards cell culture production. Because production is now within a bioreactor and not an egg, there's more control over the production process and less introduction of contaminants. Of the existing cell lines, the Vero cell line is one of the most widely used for production. This cell line originates from the kidneys of African green monkeys, and an image of confluent Vero cells is shown in Figure 4. Production of vaccines with Vero cells involves two phases, the upstream phase and the downstream phase. During the upstream phase, the virus is introduced to the cells and then grown with a perfusion bioreactor with microcarriers and fixed bed technology. This is ideal because the fixed bed technology minimizes shear stress and contact with gas bubbles. The perfusion system eliminates the need for a retention system and risk of fouling filters. Then, during the downstream phase, the Vero cell lines must be detached and retrieved because Vero cells grow adherently. The detachment is necessary but not ideal because there are limited options for process control. The detachment can be done with a substance called trypsin, and the process is called trypsinization. The virus is then retrieved with chromatography, anion exchange, or gel filtration. Vero cell vaccine production is limited by trypsinization because it is a major bottleneck for Vero cell production. While the production process is significantly less susceptible to contamination compared to production methods that require chicken eggs, the Vero cell line has a relatively low cell growth rate and long doubling time. They can also be prone to aggregation, which is detrimental to production. Now we'll move on to manufacture of mRNA vaccines. In the first step of the process here, mRNA is transcribed in vitro in a bioreactor at 37 degrees Celsius from plasmid DNA, which would encode the antigen of interest. Then, T7RNA polymerase transcribes the mRNA transcript with chemically modified ribonucleoside triphosphates. While that is happening, the mRNA is co-transcriptionally capped with 5' cap analogs. Then, in the next step here, the DNA is digested with DNase 1. Afterwards, the plasmid DNA undergoes downstream purification with the methods described here and is eventually encapsulated into lipid nanoparticles in this step here. Lipid nanoparticles are important because they protect the mRNA and allow for stable delivery to target organs. The encapsulation process is a bottleneck and is often done with microfluidic mixing devices. Then, the vaccine enters the fill-to-finish process here, where the vaccine is ultimately packaged at the end. As seen in Figure 7, storage of mRNA vaccines could require temperatures as cold as negative 90 degrees Celsius, although the more commonly used temperature is negative 80 degrees Celsius. Unopened vials can last from 2 weeks to 6 months. However, the storage temperatures are also extremely cold and range from negative 50 degrees to negative 15 degrees Celsius. Due to the nature of the storage requirements, it's very likely that some vaccines will be lost during distribution and will therefore require overproduction. Although another solution is to develop a more thermostable vaccine formulation, the amount of time necessary to develop a novel formula that would stabilize the mRNA within the vaccine is unknown. Furthermore, the vaccine would then need to go through clinical trials after development and subsequently need approval and may not be developed in time and manufactured in time to provide swift response to a Zika outbreak. Therefore, it may be more wise to focus on methods to streamline current mRNA vaccine production. In spite of the supply chain weaknesses of mRNA vaccine production, the process has a great number of strengths that make it a good candidate for potential Zika virus vaccine production. First, the production method does not use cell culture technology, which eliminates any concerns about cell sensitivity. This includes concerns like shearing of cells or removal of cells from solutions before continuing with production. And because mRNA vaccine production does not use cell culture, a continuous reactor, like an extended channel bioreactor for example, could be used for production instead of a batch reactor. However, the greatest strength of production is its flexibility. As long as a suitable plasmid DNA sequence is selected to create the antigen of interest, the same machinery and enzymes can be recycled. Therefore, the plasmid DNA that encodes the Zika virus envelope proteins could be inputted into an existing process, and the same infrastructure used to manufacture COVID-19 vaccines could be recycled to manufacture Zika virus vaccines instead. Since the COVID-19 mRNA vaccine has already been approved and has successfully been in large-scale production, a potential novel Zika mRNA virus would greatly benefit from the existing workflow. This table is a summary of the different vaccine production routes, which also includes the mRNA production limitations and advantages from the previous slides. The Strategic National Stockpile is a stockpile created by the Department of Health and Human Services to expand on nationwide response to infectious diseases. If a Zika virus mRNA vaccine is manufactured in the future, it would be beneficial to the production process to stockpile a necessary device for production. To choose an optimal device to include, it would be best to address the bottleneck of the mRNA vaccine production process. The process has already been successfully scaled up, as Pfizer already has the capability to produce over 1 billion mRNA vaccine doses. However, if production were to be modeled with a 30-liter bioreactor, which is believed to be the largest feasible size, mRNA drug manufacture would take about 2 days. Of those 2 days, 1 day would be required to finish LNP encapsulation. In order to alleviate this bottleneck, multiple microfluidic devices can be run in parallel, or a larger device could be chosen. It could be more wise to run multiple devices instead of upgrading every device in the production process to a larger size, as it would allow for adjustment to any changes in vaccine demand. To better support production, the SNS should stockpile microfluidic devices of various sizes, as different plants may use devices of different sizes. In order to suit a large-scale process, the device should be crafted from glass or metal for pressure and rigidity. An example of a device that could be stockpiled is the device shown in Figure 8. The device is called an ILINP device, and the structure as shown in Figure 8 is suitable for potential use because it has an etched design, which allows for microchannels, which are necessary in large-scale production. If the LNP encapsulation bottleneck could be successfully addressed by stockpiling these devices, then Zika virus vaccine production could increase in speed and prevent further spread of the disease. My references are included in the following slides. Thank you for your time.